structural support, and it also prevents the cells from bursting and collapsing. Now to talk about the plasma membrane, it has a semi-permeable nature and it also interacts with the outside world. And we have some specialized structure called mesosome, which we have discussed here. These are nothing but the extension of the plasma membrane, okay, and these extension can be in the form of vesicles, tubules and lamella. Okay, there are three types of form for the mesosome, and the functions are they can helpful in the cell formation, cell wall formation, DNA replication, distribution of daughter cells, respiration, secretion process. Also it will increase the surface area of the plasma membrane. Now apart from this, we have some specialized structures called chromatophores, which are seen in cyanobacteria kind of organism, and these chromatophores are contain the pigments. Now if the bacteria is motile, and it will contain some specialized structure called flagella. So the bacteria can be either motile or non-motile. If it is motile, it contains flagella, and this flagella has three parts, which is filament, hook and basal body. Now among these three, the filament is the longest structure. To continue with that, apart from the flagella, we have some structures called pili and fimbriae, but these don't help in the motility. Okay, only flagella will help. This pili are of tubular structure and fimbriae are of small bristle-like structures, and these are made up of special protein. In case of fimbriae, these are nothing but the fibers, and they help the bacteria to attach to the rock or host wherever it wants. Okay, now we will see the questions for that page. So what are the types of bacteria based on the shape? Extension of plasma membrane is called as types of glycocalyx. Okay, which part of the cell prevents it from bursting and collapsing? Which is the longest part in the flagella? This one we recently discussed. And what are the layers of cell envelope? The three layers which we discussed. I hope you are able to answer all these questions. Now let's continue with this page. Now we will talk about ribosome and inclusion body. In the previous video, we have talked about the prokaryotic cell and what are the cell envelope present and what are the modification and their function. Now we will talk about the ribosome and inclusion body. The prokaryotic ribosomes are present in the plasma membrane. So P ribosome, prokaryotic ribosome, and it starts with plasma membrane. So PP. Okay, remember that. And these ribosomes are of 15 nanometer to 20 nanometer in size, and they are made up of two subunits, which are 50S and 30S, and together it is the 70S plus prokaryotic ribosome. So whenever they are talked about the what is the ribosome present in the prokaryotic, you should say 70S. If they ask about the subunit, it is 50 and 30. Okay, not 20. Now these ribosomes are the site of protein synthesis. This is a really very important point. The ribosomes are the site of protein synthesis. And we have something called as polyribosomes or polysome. These are made when the several ribosomes, that is many ribosomes, are attached to single mRNA. Know this word. What is a polyribosome or polysome? Several ribosome plus single mRNA together form this. And these ribosomes translate the mRNA into protein. We have the central dogma, right, where we will talk about all this in the 12th chapter. We will discuss this later. So what is the function of polysome? Is to translate the mRNA into protein. Are we good with the prokaryotic cells? Okay, we have another topic here. It is an inclusion bodies. So the reserve food materials in cytoplasms are formed in the formed in the form of inclusion bodies. Okay, in cytoplasm, the reserve food materials are stored in the form of inclusion body. These are non-membrane bound organelles. So to talk about the examples, phosphate granules, synophisian granules and glycogen granules. And we have gas vacuoles, which are present in few organisms such as blue-green bacteria, purple and green photosynthetic bacteria. So with this, we are concluding the prokaryotic organism. Now we will move on to the eukaryotic. So what are the eukaryotic cells? Protists, plants, animals and fungi. So apart from Monira, we will classify all the organism into eukaryotic cell. Now the eukaryotic cells will have nucleus plus nuclear envelope, which in prokaryotic it won't be. Okay, And apart from that, we have locomotory and cytoskeletal structures in case of eukaryotic cell. Now we will see the major difference between the plant and animal cells. So the plant has cell wall, the animal cell don't have. It has plastid, which lacks in animals. It has large central vacuole, it is not there in animals. But in case of animals, we have centrioles, which is not present in plant. We have discussed already about the centriole. It will help during the cell division. We will discuss about this further later. Now we will see the questions for this page. So the questions will be, what is the size of ribosome present in the prokaryotic cells? Site of protein synthesis, which translate the mRNA into proteins. In cytoplasm, the reserve food materials are stored in the form of dash. So centrioles are present in what? Plants or animals. Several mRNA plus several ribosome is called as polysome. Is it true or false? Now we'll move on to the next page. So in the last page we have discussed about the general things about the eukaryotic. Previously we have discussed about the prokaryotic cell. Now we will talk in detail about the eukaryotic cell. Okay, now firstly we will start about the cell membrane. In earlier days, it was said that the cell membrane is made up of lipids, and these are arranged in bilayer, that is 2 layers, in which it has 2 parts. One is polar head and the next is called the hydrophobic tail. So phobic generally means fear. So hydrophobic means it is fear of water. That you can understand, right? And so these together it is called as a lipid. Lipids are arranged in bilayer. It has polar head and hydrophobic tail. So these are arranged in such a way that the polar head connects with the outside water or the cavity, and the hydrophobic tails are saved. Okay, this is the specialized structure of it, and these lipid component of the cell walls are mostly contains phosphoglyceroids. Okay, this is an important point. The lipid components of the cell wall mostly made up of phosphoglyceroids. And after some times it is known that apart from the lipids we have protein and carbohydrates also in the cell membrane, and the amount of protein and lipids are different in each cell. To talk about in RBC, we have 52% of protein and 40% of lipid. Okay, now depending upon the ease of extraction, these membranous proteins are divided into 2 types, which are peripheral and integral. Peripheral means it lies on the surface of the membrane, so it can be easily extracted, right? And the another is called integral, which is partially or totally buried. Now after these discoveries, there is an important discovery done by Singer and Nicholson in 1972. They called it as the fluid mosaic model. This model says that there is an quasi-fluid nature for lipids. Okay, the lipids have a quasi-fluid nature which helps in the protein's movement. This quality, or it is measured as fluidity. Okay, the quasi-fluid nature of lipid is measured as the fluidity, and it helps in the protein movement. And one of the most important function of the plasma membrane is the transport of molecules. This transport of molecules can be done in different terms, either passive transport, in which no energy is needed. It will be simply transported to one place to another. Okay, and we have simple diffusion. This is done for neutral solutes. Simple diffusion is when it is moving from higher concentration to lower concentration. It is easy, so no energy required, and it is called as a simple diffusion. So same when from higher concentration to lower concentration, water is moved. Okay, in case whenever the molecules are moved from higher concentration to lower concentration, it is called as simple diffusion. But if water is moved, it is called as osmosis. Okay, movement of H2O by diffusion is called as the osmosis. Now active transportation is where the energy is required, because here it will move from the lower concentration to higher concentration. Generally, if you assume putting a thing from higher to lower is easy, but making it towards the high, it is tough, so it needs some energy. So this is called as active transport, and the example will be sodium potassium pump. Now we will talk about the cell wall. We talked about the cell membrane. Now we will talking about the cell wall. These cell walls are non-living. Cell walls are non-living and rigid, solid kind of thing, and it covers the plasma membrane in plants and fungi, because cell walls are absent in animals, as you know now. Now we will see the cell walls are made up of different types of components in terms of different kinds of plants. In terms of algae, it contains cellulose, galactans, mannans and calcium carbonate, which is a mineral. But in case of other plants such as pteridophytes, bryophytes, these are the other plants, right? It is made up of cellulose, hemulose, pectins and proteins. These are the different types of component present in them. And we have middle lamella, which separates the which glues or connect the different neighboring cells. With help of different neighboring cells, with the help of middle lamella, the different neighboring cells are connected together. Okay, this is the use of the middle lamella, and it is made up of mainly calcium pectate. And between the cell wall and middle lamella, okay, what is the middle lamella? Which connects the different neighbor cells. So between the cell wall and middle lamella, we have plasmodesmata, which connects the cytoplasm of neighboring cells. You are clear with these terms? Middle lamella neighbors the cells together, and this between cell wall and middle lamella we have plasmodesmata, which connects the cytoplasm of neighboring cells. Okay, now we will see the question for this page. The first question will be, in the lipid bilayer, which interacts with the outside world, outside cytoplasm. What is the percentage of protein in RBC? Fluid mosaic model I have given in short form like FMM. Fluid mosaic model was found by whom and in which year? Give an example for active transport. Pectins are found in algae cell wall. Is it true or false? Which separates the cell wall and the middle lamella in eukaryotic cells? I hope you revised this page. Let's move on to the next.